I think everyone knows about the pandemic plaguing the planet, COVID-19. The pandemic that brought all of us into our homes, the one that rattled society, the economy, and so much more. Humans have put up a good fight against this pandemic. More and more people are getting vaccinated. Things are starting to look up now. So let's get into details and look at what started it all. This is the virus behind the pandemic. Pretty small, huh? How did this new virus cause the planet to go from this to this? A virus is a mischievous little creature. Our bodies are designed to prevent a virus from attacking us, and this works most of the time. In some instances, these little buggers find a way to overcome at least one of the obstacles the human body creates. The entire process by which a virus infects the body takes numerous steps. I will be focusing on one crucial step, and that is translation. First of all, what is translation? Translation is essentially the formation of a polypeptide from messenger RNA. Messenger RNA, or mRNA for short, is kind of like a string containing a code of instructions. The ribosome, which is sort of like a polypeptide factory, reads this code and subsequently makes a polypeptide. So how does this teeny tiny virus shut down such an important process? There are three main methods, but before I get into that, let me introduce you to two proteins, EIF4E and EIF4G. How about I name these two dudes, just for the sake of simplicity? How about Ed and Greg? Perfect. Moving on. Ed is a cap that the mRNA wears on its 5' end. It signals the ribosome to come toward the mRNA. However, for the Ed protein to actually make this signal, it needs its buddy Greg. Translation cannot happen if the two proteins aren't with each other. When these two attach, translation finally starts. Now, onto the three methods. The first is cutting the two apart. It's pretty self-explanatory. A virus simply cuts the two proteins apart if they've already bound to each other. Ouch. The second one is called dephosphorylation. As the name suggests, it's the removal of a phosphate group. Think of this phosphate group as Ed's key to binding with Greg. If the phosphate group key isn't present, then those two cannot join together. And if they cannot bind with each other, then translation cannot happen. In the third method, we have a new player in the game. Its name is 4EBP1. Again, for the sake of simplicity, I'll name this protein Chad. Chad is this macho protein capable of stealing Greg's buddy, and that's exactly what happens. A dephosphorylated Chad protein binds to Ed, leaving Greg all alone. So the virus has achieved its goal. It successfully stopped cellular translation from happening. Now what? The virus takes over the process of translation. Essentially, the host is now infected.